All right, I'm Dr. John Catanzaro. I want to welcome you to the Health Coach 7 Q&A. Today's topic is genes and methylation. And before we go into the meat and potatoes of questions, I just want to kind of go into a little bit of a synopsis of what we're looking at when we talk about genes. Really, really important. You know, genetics has gone really far to where now we can actually take a genome from one species, and we can insert a genome in one species and go into another species. So in other words, you can take a nucleus of the cell, take the genome sequencing out, digitize it, and then use that sequence to create another genome into another species. And so now they're actually figuring out ways to where they can build chromosomes, and they've actually built a chromosome. They don't know what all the gene segments do on that chromosome that they just synthetically built, but we're not doing that type of genetics. We're doing something a bit different. We are doing, how does our human genome behave? Okay, really important. So now I just want to give you a few basics of the human genome. There's approximately 23,000 genes plus in the human genome, and there's about 15 million SNPs. And then when we look at the genome, the SNPs are basically single nucleotide polymorphisms. And what that basically means, it's a typographical error that happens along a gene's path that we're looking at in a particular gene, or it could be between a gene's path. So now when we look at SNPs, okay, and when we talk about methylation and SNPs, there's various types of SNPs that happen on specific genes that will affect methylation. So now what we did is we broke methylation down into categories because the different systems of the body can affect methylation. For instance, genes in general will encode for methylation. And if you look, I'm going to share my screen with you just right now so that you can see what I've outlined here is on gene cards. On the left side, you actually have a sample plan that what Health Coach 7 puts out in personalized genetics. So this is a personalized action plan that we develop based on the genes. So it goes into a little bit of the introduction of genetics 101, talking about chromosomes, talking about DNA, which is the building block of chromosomes and genes. Okay, and then it goes into categorizing, where it's found, where the genes are found, what is a chromosome, what does a gene look like, what is it going to encode. Basically, we're looking at genes encoding proteins. And there's various types of proteins of the body, right? So now when we look at this, we're breaking it down in dynamics. And of course, genetics is the first dynamic because genes are controlling pretty much all of the information, that your protein encoding information of your body. So what we decided to do is to break it down into the component parts of methylation and metabolism, methylation and immunity, methylation and detoxification, methylation and hormones. We also have subcategories like methylation and SNPs, methylation and allergy, methylation and I think that there was one other topic. I forget what it was. Do you remember? There's a bunch of topics here. So we're doing the dynamics first, and then we're going to break it down into SNPs, allergy, which will cover histamines and things like that. And you're going to find this very informative because methylation is a very, very complex process. So this is the left-hand side. As you see me scrolling, then we talk about epigenetics and what epigenetics is. And basically, epigenetics is the programming of what they call favorable programming of your genome specifically. So if we look at it, we want to change bad signals into good signals, right? So how do we do that? Well, we have an intelligence program in epigenetics. We can actually create an epigenetic intelligence that would actually address what they call susceptibilities or sensitivities of the genes. And when we look at these gene SNP combinations, or what they call variants, what we're really looking at is what are the strong points and what are the vulnerabilities. So that's why we plot it in sensitivities and susceptibilities. Very important. Just because the genome might show susceptibility in an area doesn't mean that a person is doomed forever. because you can actually overcome susceptibilities with favorable signal changes. And that's what epigenetic intelligence is all about. Basically, how do you do that? Well, we're going to talk about it in several of these segments. We're going to talk about favorable things to do to change the potential bad signals into good signals to help the sensitivity susceptibility of the genome. Okay? Basically, you can't change the genome. The genome is what it is. If you are a zebra with black stripes and a white background, you will always be a zebra with black stripes and a white background. You'll never be a white striped zebra. Okay? That's basically the bottom line. So you're not going to change your genome, but you can certainly change the signals, the epigenetic signaling of that would assist the genome into becoming a good, functional, healthy genome. So as you can see, epigenetic mechanisms involve several different things. As we scrolled here, you can see that development obviously is the first part, but there's environmental influences, drug and pharmaceutical influences, aging influences, dietary influences, of course, exposures. All of these different things have various type of effects on epigenetic mechanisms. But now the important thing to remember here is that when we're talking about epigenetic signaling, we are talking about influencing DNA methylation. And the very basic part of methylation is when we talk about methyl groups, which is an epigenetic factor found in the genome, and they're known as CpG islands. Okay, a CpG island is what you're seeing pretty much taken out here. This histone tail and this histone group. They're known as CpG islands, and CpG islands are where the influences of methylation occur. And methylation basically and simply is the genome's ability in dictating how that cycle is going to go. So like in other words, your methylation cycle is determined by your genome, and if you have susceptibility and sensitivity, you might not necessarily be the best methylator in keeping the balance of methylation, okay? But your epigenetic factors can profoundly influence the direction of methylation because methylation is there as a normal process of DNA regeneration. So when you think about it, just think about DNA as being that wonderful molecule that's so perfect that it could be perfectly good or perfectly bad. So in other words, in cancer cells, DNA is not perfectly good because DNA is actually encoding for negative proteins to where cells are misbehaving and they wind up becoming damaging cells, right? Well, in healthy cells, the DNA methylation cycle is pretty much intact. And what we want to do is we want to be sure that we're keeping the DNA regeneration ability in a healthy fashion, and methylation is that ability to keep healthy DNA regeneration. So basically, methylation is the process by which your DNA is protected, okay? Protected from harmful influences. Now, when we talk about methylation, we're talking about hyper, which means aggressive, or over methylation, which can be influencing a lot of different things. And when we talk about brain and methylation, you're going to learn how various type of methylation changes will affect the brain. And we're going to be getting into, the methylation diagram in general when we're going to start going into the various points of methylation cycle, which involves several different gene controls. And I don't have that up on the screen right now, but what I wanted to do is I wanted to show you the number of genes that are involved in methylation process. Right now, if you were to do a search on gene cards, which I have up on the right side here, you can see this area right here is genecards.org. When you actually type in methylation support, the methylation support will actually pull up all of the associated genes that are involved with methylation support. There are approximately 906 of those genes currently known that are coming up with methylation support. And of course, MTHFR is in there, as you can see, right? And then there's MTR, which a lot of these genes you can remember on your conversion reports. COMT, which is the catechol-methyltransferase, and there's you're going to see the BHMTs too. It's in there somewhere in the pool, and you can even see a lot of other ones like MAOB, which we actually know is controlling histamine responses, and estrogen receptor, which is controlling estrogen responses, and monoamine oxidase A, which is a main neurotransmitter to help with regulatory transmission. I mean, it's pretty amazing that you have about 906 genes that are responsible for methylation specifically. So now that's why I want you to understand that we don't want to get too excited, overexcited with SNPs, because when a person gets really excited about just MTHFR in that mix, they discover that they are coming up short because there's a whole lot of other genes here that are responsible for keeping good methylation cycle, and as we're going to be seeing. So I just wanted to point that out. I think some of you may have remembered, if you actually go to you know, you can actually search this out yourself if you go to the internet and you open up, let me just show you right here. Just go under the methylation cycle. If you type that in, you're going to actually come up with various types of images that you know, a lot of you probably recognize on some social media. So if you look at some of this here, some of it could be quite confusing. But let's say if we open up that one right there, some of you can actually see that, you know, of course, you see the MTHFR C677T. Some of you may have seen Amy Yasko's hypothesis here. Hers is not the only one that's out there. There are a lot of authoritative text ones that are out there as well that you can compare to. She does a fairly good job in trying to outline that, but there are some things that are not included in her methylation pathway. So I would probably be using more of a what they call biochemical text to look at it. But just looking at Amy's, for instance, you know, you can see you've got a lot of MAOBs, a lot of COMTs, a lot of MAOAs, a lot of NOSs, a lot of BHMTs, HCY, CBSs, the ACEs. A lot of these you recognize on your conversion reports, right? And they are all important because each one of these particular cycles within the methylation cycle are responsible for a lot of different things. Like, for instance, the ammonia metabolism pathways, the methionine catalyst pathways, which include SAMe, with the actual ferritin shuttling pathways that actually include NADH, which are known as the end of S's. So end of S7, end of S3. These are all important gene controls that are going to help regulate, and you're going to find them in that methylation support list that I showed you just a minute ago. So I want you to just keep in mind that methylation is not just about MTHFR. Methylation is about cross-related control balance in these genes to be sure that systems and system integration is considered. So when you're looking at your genes, please do not isolate just one gene out. Please do not isolate one SNP out. Please understand the relevance and realize that SNPs are basically non-consequential in 97% of the time. And so with that, I'm going to open up the floor now for questions so that we can actually begin that process. And I'm sure that a lot of you have questions regarding methylation in general. So let's go ahead and take some questions. And I'm just going to open up the floor here. So feel free to ask any question here. Just raise your hand for a question. I see a couple of raised hands right now. Is it okay to mention names or not? No. All right, so we're trying to figure out who's going to, hold on a second. So we have several people that are raising hands right now, and we can't tell who raised their hands first. This is kind of interesting. So I think, I don't know if we can, I don't think we can mention names on the webinar. So what I'm going to do is I'm going to ask you to write your question down for me. Let me just try something here. Does everybody have their sound on? Someone say something for me. You're going to get really bad. I'm getting too much feedback. So. All right. Bear with us while we're trying to figure this thing out here to get these questions asked. So what I'm going to do is I'm going to ask you to write your questions out. There's a little question box right there. Type your question out for me and let me know who you are. I think when you type the question, it's automatically going to plot who you are. All right, so the first question that I have here is, you said SNPs don't create problems. Why are problems listed under SNPs in general forums? So basically, to answer that question is, when SNPs are actually being looked at, single nucleotide polymorphisms, they're looking at what they call world population. And if you actually went to NCBI, which is a national database, it's going to be actually plotting SNP and the SNP significance. And there's an allele, what they call a minor frequency allele, that's plotted. And the alleles are usually associated, if they're minor, with some type of a risk possibility. So for instance, if a person actually has MTHFR, there may be a problem with, for instance, folate metabolism, which might include issues with homocysteine, which can cause more cardiovascular risk, et cetera, et cetera. So when you look at that, there is the possibility that it might be associated with a risk, but a large part of the time it's not. Now, what we find is when we actually see related genes like MTHFR and MTRR and some of these other related methylation genes working together and showing high possibility of sensitivity, then we might see it validated on a person's personal history and also on some of their lab results, their lab testing. So their lab testing might show elevated homocysteine, for instance, or they might show an elevated high sensitive C-reactive protein, which indicates a lot of inflammation in the vessels. I mean, we're going to be seeing patterns like that, okay? So SNPs are not always associated with disease frequency, but they can be. A large part of the time they're not, and sometimes they may affect the expressed genetic expression, but most of the times they don't. All right. So the other question is, why has so much focus been made on MTHFR? Well, because a lot of the times MTHFR is really strongly connected with a lot of what they call mood disorders and mood changes. MTHFR and autism, MTHFR and schizophrenia, MTHFR and anxiety and depression. MTHFR is, in fact, methylene tetrahydrofolate reductase. And if you say that real fast, it's a tongue twister. But methylene tetrahydrofolate reductase basically is an enzyme that's responsible for helping the metabolic movement of folate. And if you actually have a problem with that MTHFR pathway, you're going to have associated pathway disturbances that include some of those neurotransmitters that depend upon that methylation movement that MTHFR is a command gene of. Now, MTHFR is not the only gene that controls that methylation process, so it's unfair to hinge everything on MTHFR. And a lot of the times you can see some connections with increased risk of cancer with MTHFR expressions, and there's been a lot of controversy over C677T and A1598C and some of the other ones. They're looking at it and they're saying, you know what, I think there's way too much stuff attributed to MTHFR, and I think we're looking at it the wrong way. I would say that MTHFR is an important gene association with gene SNP variations, but we don't want to be just isolating it out itself, because there are other influences on neurotransmission. For instance, like the COMT and people not being able to tolerate methyl donor products, and BHMT and CBSs. Those all have influence on that methylation cycle and indirect influences on MTHFR's performance as well. Okay, so that's very important that we keep that in mind. Another question is, you briefly mentioned that methylation affects histamine. Can you expand on the relationship and how to address histamine intolerance and mast cell issues? Can you do anything to support methylation to decrease histamine? Now, that's a very, very good question. Did everybody hear that question? Please raise your hands if you heard that question, because I can repeat it again. Just hit. Very good. Majority of you have heard that. So the question is methylation affecting histamine, and how so, and what can we do about it? Yes, methylation can affect histamine, and histamine can affect methylation. So bottom line is that histamine imbalances If you're going to look at it the other way around, you don't want histamine to negatively affect methylation, because the methylation cycle is basically it's a process by which other systems can alter its process. So, like in other words, if you actually have a lot of inflammation going on in your body, common sense would tell us that methylation is going to be affected in some fashion or another, but caused by that methylation, that inflammation. The same thing with antigen processing. Now, say for instance you were eating a food that you chronically react to. Maybe it's a food that you're having what they call a delayed hypersensitivity response to. If you're having a food that you constantly eat that you don't know is provoking a what they call antigenic response, where an antigen is formed and an antibody is made to the antigen, naturally, as a consequence of that, there's going to be some histamine formation surrounding that. And with chronic activity like that, that might be an aggravated movement into what they call mast cell activity. Now, the thing about mast cells is that mast cells clear the circulation, clear the blood very fast, and they hide in the tissues. So, like in other words, you can have chronic inflammation associated with chronic mast cell activation, and mast cells are known as macrophages, so they actually provoke inflammatory responses. Is that going to provoke histamine responses and ongoing histamine? Yes. So you have histamine, mast cell, more histamine, more histamine, more mast cells, more antigen provocation, more mast cell potential. So people that have what they call chronic mast cell activation are probably having chronic hypersensitivities as well, okay, which we see as a commonality. Now, in the brain, histamine acts as a neurotransmitter. So if you have depletion of histamine in the brain, you're going to show more hypermethylating patterns in the brain, which can actually create more disturbances with the neurotransmitters like dopamine, GABA, serotonin, the hormone melatonin as a commanding hormone for cycling. You're going to find a lot of these relations. Serotonin is another one of those. They're going to be disturbed by the imbalance of histamine in the brain. The same thing with glutamine. Glutamine in the brain is not a favorable item. If you have excessive amounts of what they call glutamic acid in the brain, it creates what they call a major excitatory effect on neurons, and it causes a paralysis to occur in some neurons. So you can actually see you've probably heard of with GAD1, glutamine decarboxylase 1 enzyme. They have something called the stiff man syndrome or something, paralysis and seizures. The reason why these things happen is because glutamine is a toxic agent in the brain, whereas in the gut it's like a food. For athletic performance, it is like a real strong, favorable biochemical to use for athletic performance. But if you actually have a problem with cyclical GAD1 and neurotransmitter maladaptation and histamine depletion or histamine excess, you're going to have methylation disturbances in the brain. So brain hypermethylation and hypomethylation are not favorable. They're not favorable type of cycles in any of the systems, but the brain is a lot more sensitive to those changes. And that is the reason why this brain gut interaction thing is so important to look at, because any toxicity in the gut is going to affect the neurotransmittability in the brain. Your neurons will be affected by toxic agents that cross the barrier in the gut, because they're going to cross the blood-brain barrier as well. So you got to remember that. Okay, that's very important. Excellent question. I mean, that histamine is a big deal. The next question is, what is the relationship between methylation and autism in children? And methylation and autism, that's a very big topic in autism. Currently, when we look at autism, it has a lot of components when we're looking at it. Some mitochondrial disturbances associated with cell energetics, cell-related fatigues, but mainly when we're looking at mitochondria, cell energetics, we're talking about across the board, how does this affect the individual neurons and the ability for the neurons to transmit? So methylation can be an aggravating, imbalances in methylation can be an aggravating factor in poor prefrontal clearance, for instance, in the dopamine receptivity. So if you look at dopamine receptor 2, DRD2, and if you profile DRD2, okay, for instance, I'm going to go look it up right here, DRD2 relationship. You see that on the screen now. I just changed it for you. Please write down your questions. You can just type them in, okay? Anybody that's in the seminar right now, please type your questions in. But let's go to DRD2, dopamine receptor 2. DRD2 is seen if you have poor prefrontal clearance. DRD2 is one of those genes that's listed, gene SNP variants that's listed in the methylation support pathways. And if you look at DRD2, we're just going to kind of broaden this up a little bit so you can see it better. The DRD2 is responsible for dopamine metabolism in the brain. And if we look at it specifically, we're going to see where it's located. It's actually located on chromosome 11, and it's located on the long arm of chromosome 11. The P arm's the short arm, the Q arm's the long arm. That's red dash is where you're seeing where the gene is located. And the gene is 66,097 base pairs long. So remember when I talked about SNPs and how many possibilities can SNPs follow? I mean, you got 66,097 possibilities right there that a SNP's going to insert itself in that base pair path of that gene. How they come up with it along the chromosome is they'll take this end point from the start point, and that's how the number of base pairs they come up with the gene. So that's how they determine that. And then when they're looking at it, well, how many amino acids make up this gene? There's 443 amino acids that make the gene up, okay? And when you're looking at diseases and disorders, you're going to find there's a whole big laundry list associated with diseases and disorders. And autism is part of this process, and not just schizophrenia and alcoholism and Parkinson's and some of these other neurodegenerative disorders, but autism, autistic spectrum disorder, and DRD2 is also present. You're going to see that with various other ones, with not just dopamine, but you're going to see it with serotonin. You're going to see it with GABA. You're going to see it with a lot of these other neurotransmitters associated with that, keeping that balance. So methylation, poor methylation would aggravate more autistic spectrum type signs. So if you have a methylation disturbance occurring, you're going to have more aggravated type of signs associated with it. And then there are ways of being able to program those neurotransmitters so that you have less hyperreactivity or more expressive, what they call inhibitory action, and maybe affect the neurotransmitters so that they're not so hyperexpressed as you would find in a lot of autism. So dopamine and a lot of the other neurotransmitters are involved there. As we actually move along, I wanted to just show you that dopamine, when you look at the cell expression of it and where you find it, this particular gene SNP variation in cells, and especially in neurons, it's very confluent in the cytosol. So you're going to see a lot of it being produced in the interior of the cell, which is really important to know. Okay, so I hope that answers that question with that autism. Autism is a very difficult disorder to characterize, but methylation is definitely going to profoundly influence it either to help correct some of the hyperexpressed symptoms of it or to address, some of the manifestations that are unfavorable, and then to try to create a better balance. Autism is a tough one, but that's a very good question. Does anybody else have any other? Okay, so another question is, can methylation affect sleep? Yes, I mean, methylation is a profound sleep. When you're looking at sleep, methylation, when you're talking about its influence on GABA and influence on the circadian clock, like Cry2 and Timeless, some of these genes that are responsible for and the PER1 and ARNTL, a lot of these genes are responsible for keeping a 24-hour rhythm. And when we find that there's a lot of what they call a hyperexcitatory behavior in the neurons, we're going to find that sleep cycles are altered pretty dramatically. And hormones affect that. So for instance, the adrenal pathways will affect that. If you have a real bad adrenal, what they call hypersensitive adrenals, or you have adrenal-related fatigue, or you've got definite imbalances between thyroid, adrenals, and reproductive hormones. These hormones dramatically affect how you metabolize certain type of neurotransmitters, and you're going to have a bottom line effect on sleep. So estrogens can affect sleep, thyroid hormones can affect sleep, melatonin depletions can affect sleep, imbalances between GABA and serotonin and can affect the sleep cycles. I mean, so you're going to see a lot of those related elements of methylation affecting it. And bottom line is, if you're looking at MTHFR and you're looking at MTRR and you're looking at some of these other COMTs, you might find that when you're addressing methylation and some of the surrounding causes of methylation disturbances, like maybe gut associated or immune associated disturbances, once you clear up those areas, if a person's having chronic, for instance, exposure to grains, and they have a terrible grain intolerance, and they're having cross-reactive antibody responses, and they're getting a lot of antibody antigen complexes forming going across the blood-brain barrier, then of course they're going to have problems with sleep, because those antigen antibody complexes will actually inhibit healthy neurotransmission, because those antibodies are responsible for blockade. They will block healthy neurotransmitter signaling. So methylation, of course, will be corrected by addressing some of these other exposures, and then trying to also at the same time look for the holes in the methylation cycle that you're seeing according to the genetics, maybe the susceptibilities, and then addressing those as well to help balance out the methylation. So anyway, I hope that there's a lot of detail when it comes to assessing sleep and methylation. So we actually are looking at many areas of focus. Does anybody else have a question? Please feel free to answer a question. There's no such thing as a question that doesn't make sense. Okay, so I mean, I have people that ask questions that provoke a lot of thought, and we can, you'd be amazed at some of the answers that come from that. So please feel free to ask those questions so I can answer them for you. Anybody else have a question? Just type it out. Can you still hear me? If you can still hear me, raise your hand. Great. So I'm not seeing any other questions there. Do you know how to, what was that? Okay, here's a question. What are some of the top nutrients and supplements that create a healthy methylation system? All right, so now a lot of you probably are aware that methylated folates, like the quadrafolates that are out there, the methylcobalamin, which is methylated B12s that are out there, SAMe, CoQ10, NADH. These are all profound mitochondrial regulators as well as methylation regulators. You've got magnesium. So, for instance, people that come up with deficiencies of magnesium, if you have red cell magnesium shortages, or you find that you come up with a TRPM6, that gene right there, if you actually had the TRPM6, is a gene that's associated with magnesium deficiency. And magnesium is a big player in methylation balance. So if you actually have deficient magnesium, you're going to have issues with methylation imbalances, because magnesium is part of that methylation cycle and every biochemical process that's involved there. So magnesium replacement is important, as well as CoQ10 replacement is important. P5P, which is vitamin B6, is very important. So these are a few of those nutrients that, now it's important to know that you just don't want to be taking a methylated product if you don't know where your other systems are, because if your system's around, like your immune system or your metabolism is altered, or your hormones are not in correct balance, they might be provoking methylation changes. So you really want to be addressing things intelligently, because remember, we are trying to create epigenetic intelligence by identification and solving those areas that might be creating that load, okay, on the methylation cycle. So we want to be sure that uh, we address that. Oh, that's an easy question. How can I see you? Basically, if any of you are interested in seeing me and consulting with me, you can actually go on our website at www.healthcoach7, all one word, with the number 7.com, and you would be able to set up a consult with either myself, John Catanzaro, or Dr. Jeff Thomas. Both of us are qualified in the area of personalized genetics and medicine and functional medicine. And we would be happy, I would be happy to look at your 23andMe raw data and your history and your existing labs that you have and put together a personal action plan for you that is actually making more sense. So it actually helps to foundationally move you in the direction you want to go. We also team up with your practitioner that you have at home. We can team up with them and clinically interface with them in case you needed a certain test order or you needed some more interventions that might include things like intravenous replacement. Some people have to do IV glutathione. Some people have to do maybe light methylation or what they call detoxification of heavy metals. I mean, we can coordinate all of this with your and team clinically with your doctor as well. So you can go on the website and set up a consult, and all the details are there, the pricing and what you get for the investment that you make. And I can tell you that I've been consulting with people all around the world and having some tremendous results. People are just really loving it. A lot of people, the common comment back to us is that I wish we would have seen you first, because I would have, avoided spending tens of thousands of dollars on things that I shouldn't have maybe, done. And of course, hindsight's 2020 for many of us. But the thing is that when you're doing your genetics, you're a lot closer to having a personalized plan, a true personalized plan. And this is not just an advertisement. This is more of a looking at it from the standpoint of what I have 20 years of experience working with cancer and chronic illness and autoimmune disease. And I can see very strong relationships in having your personalized plan formed for you and having some intelligence put into it. So the whole idea is better adaptability, epigenetic intelligence by all means to change those susceptibilities and getting on a firmer course for your health. So I'd be happy to see anybody that's wanting to schedule a time consult. Again, www.healthcoach7.com. Okay, now the a couple of people that I know have been seeing other practitioners, and other practitioners have made recommendations for them. And I have a question saying that I've been seeing this one practitioner and need more help. So the thing is I do that a lot. And I don't ever minimize the practitioner that you've seen as saying that they haven't in any way contributed to maybe assisting moving the process forward. But what I do say is that there's a lot of confusion out there with regards to knowing exactly how to relate these genes from a relational standpoint. Remember, I said no gene stands alone, right? You got to look at these genes from a cross relationship, because if you look at these gene cards again, I'm going to show you on here on gene cards. Say, for instance, if we look at the ATG16L1 gene, for instance, I'm just going to pull that up here. I'm going to click on it. See, I'm clicking on it right now so that you can see how to use this database. But on the ATG16L1, this gene is known, if you see it on your conversion report, to be associated with inflammatory bowel disease, okay? So as you see, defects in the gene are known to be cause susceptibility in IBD type 10. But now, as you go along here, the demographics, where it's located on the chromosome, how big of a base pair, how many base pairs it is, where your SNPs can insert themselves, et cetera, et cetera. But if you go and find out, here's where it's found in the cell, but you want to know what other genes are associated with it. Now, you see this little diagram right here. These are cross-related control genes. So if you click on that, it's known as the string network of genes. So you don't want to just be isolating one gene out. You want to understand how genes interface with each other, right? Because gene interface is a big deal for keeping general cell health, remember, each one of these genes have the ability to create a functional protein. So now when genes are creating functional proteins, as you can see, we have a stellar relationship here. So if you actually look at it, I mean, if any of you are astronomers or physicists or astrophysicists, you can actually see that the expression line here is showing an interdependence in one way or another, indirectly or directly, either by stimulating, initiating, suppressing, having direct action upon. You can do the research yourself. You can actually look at each one of these genes to see the effect on the other gene. But the center of the universe right here is the ATG16L1 gene. So now if we click on the ATG16L1 gene, remember we're talking about a gene that has influence in creating inflammatory movement in the gut. Okay? So, but you've got other genes that might be affected by its lack of performance or its overexpression or its, dysfunction. And if it's healthy, great. If you have a healthy gene there, then it's not an issue. But no gene stands alone. Okay, that's what I think you need to know here is no gene stands alone. And you always got to remember that. So the ATG16L1 gene is one of those genes. Let me see, go back to, if you want to see the diseases and disorders associated with it, it comes up as IBD-10 and Crohn's and colitis. And a lot of people that in the audience might be knowing somebody that suffers with that, and this particular SNPs that are identified with this. You might actually have, like the #1 article here is saying the genome-wide associated scan of non-synonymous SNPs identified in susceptibility variant for Crohn's disease and ATG16L1. So you might find that the SNPs in the Japanese population is different from the SNPs in the Caucasian population. So when you go to NCBI, you might find that some SNPs might be positive in the Caucasian population and negative in the Asian population. Okay, so that's all based on NCBI. So you got to be sure that you're looking at these genes closely. But bottom line is it's the performance of the gene that counts. So ATG16L1 is one of the major factors in inflammatory changes in the gut, right? Very, very interesting stuff. I mean, I'm just amazed all the time when I go through this stuff. I'd be happy to work along with the doc that you're working with to look at your 23andMe data more closely, plot it in the dynamics, and then address the functional cross relationships of the genes like I'm saying here so you get the full philosophy of how I approach it. Question was asked, can you talk about the alleles and their relevance? Okay, now that's a very good question too, because in the national database, which is known as NCBI, so if you typed in NCBI and go to Google and do that, and you typed in, and you look on the right-hand side in the directory, and it says SNPs, okay? You can type in any SNP of concern, and it's going to point out what the major and minor allele would be. Now, generally speaking, the major allele is not associated with any disease significance, okay? So when we look at major alleles do not have major consequences. When we look at minor alleles, the minor allele basically is the smaller percentage of the population that is actually demonstrating what they call the possibility of having a risk allele. That simply means that there may be an associated disease risk with that particular SNP and that allele that's present. So not all of the time, but sometimes that might be the case. And the way you tell that is, a lot of the times is based upon symptoms and signs, based upon relevance in labs, based upon family history. And so you can see a lot of the relationships that way. So alleles and their relevance, if you go to NCBI, you can learn a lot about alleles and their relevance. Remember, the major allele is of no major consequence. The minor allele is associated with risk, but a lot of the times the risk that is there is usually non-consequential. Hope that makes sense. Well, then you say, well, if it's non-consequential, then why should I look at my genes? Well, because the genes in general, when you're looking at your genes are pretty much giving you the susceptibility and sensitivities of what might be possible weaknesses that you're demonstrating that might have been passed down to you by your previous generations, your mother and father. For instance, like mitochondrial genes are basically maternally inherited. There's 20,000 genes that help them, that actually command the mitochondria. The mitochondria is the powerhouse of the cell. So if you have no power, you have no energy, right? So the mitochondria helps with that power regulation. And they're maternally inherited. So it's 20,000 genes with about 39 of them that are command performance genes of the mitochondria, all from your mom. Pretty much amazing stuff, isn't it? Well, I'll be happy to work with you in assisting you with your genes, and I'm looking forward to doing that. Anybody else have a question? Anybody else? Raise your hand if you have a question. Question, you can say it. Just I want to see if I can get the sound here. So anybody with a question, raise your hand, and I'm going to click on the sound, if I may. Nobody?
But the
Anybody?
action steps are there, and the recipe guides and so forth.
Okay, can you repeat that again, please? Just repeat it again for me. Hello?
Yeah, that's on under part one in the drop down box. It says getting started,
Oh,
and that's like the first thing to do.
are
It
you?
just kind of tells you, do this, and it might say something like watch this video, and but it will give you more direction. So rather than having to go through all the screens, it's right there in front of you in paper if you print it out.
It's funny. All right, is everybody still there? Raise your hands if you still are present. Okay. All right. It looks like we're running out of time here, and I wanted to thank everybody for attending. We will be actually having this link up so that you can actually listen to it again. And I know we talked about a lot of subject matter here, and hopefully it'll get you at least primed. I did come up with a webinar last week, or I think it was 2 weeks ago, and it actually lays the foundation of genetics. Today's subject was genes and methylation. Next week is going to be metabolism and methylation. That's a very, very important subject. And metabolism and methylation, it's a huge influence. I'm going to show you the metabolic map next week, which is an intense map of human metabolism to show you how methylation can be affected by human metabolism. So anyway, look forward to that next week. Please sign up. We had some good attendance today, and I look forward to seeing you next week. And thank you for your good questions, and I look forward to working with some of you. Feel free to contact us anytime at www.healthcoach7.com, info@healthcoach7.com, and we'll be there to answer your questions for you. And then if you have questions on social media, feel free to ask when I start posting. Don't hesitate to ask me those questions. I'll be happy to get to them. I won't be able to answer all of them, but I'm sure I can get to most of them. All right. It was a pleasure talking with you today.